Well, thank you all for being here. By the way, this is our first CBAC seminar of this
fall festival.
So we have a very distinguished visitor for that occasion. Brian O'Rourke. I've known Brian since a memorable dinner in Haifa. These are many years ago. And Brian received his Ph.D. from Thomas Jefferson University in Pennsylvania, in Philadelphia. And then went and continued his both training and research activities in Eduardo Marmon's lab at Hopkins, and he stayed in Hopkins. And he is now professor in medicine, division of cardiology. Brian has done really remarkable work on using many strategies and many approaches to integrate cardiac cell metabolism with calcium cycling and electrophysiology in sort of a comprehensive way at the cellular level. And some of the work that he's done on ion channels in the mitochondria and so on has really pioneered a whole new way of thinking about arrhythmias in the context of heart failure. So Brian, it's your stage.
Thanks, Yaron, and thank you everyone I met with today for taking the time to share the fantastic work that you're doing. My lab, as Yaron mentioned, we try to look at the big picture in the cell and integrate energetics and EC coupling and calcium handling all together. But the essence of what I'm pursuing is trying to understand how the normal EC coupling, the electrical signal is coupling to the calcium release from the SR and the energy demand is linked to stimulation of energy metabolism in the mitochondria. And more importantly, in disease states, how this process can go wrong and you can get dysfunction in the energy system that leads to not only the activation of metabolite-sensitive channels in the sarcolemma, but also to oxidative stress, which impacts all of these subsystems, calcium handling subsystem, the electrophysiological substrate, and the contractile element. So what we're dealing with in this talk is the steps that I think contribute to calcium overload and arrhythmias and cell death in the context of heart failure. And from the point of view of the whole network that makes up the heart, the connections between the cells make up the syncytium, the crosstalk between mitochondria inside the cell makes a network of mitochondria that respond to each other. What I like to think of heart disease as is a cascade of failures that scales from the organelle level up to the organ system. And there are many common things in physical systems and in natural systems that can lead to catastrophic failure. And usually it's a bunch of nonlinear elements that are connected together, and often they behave in unanticipated ways, so-called emergent properties. And I think of the mitochondrial network sort of like the power grid, like in the United States. In 2003, there was a massive blackout from Michigan to New York, and 50 million people lost their power. And it started in Ohio with some relays that led to a cascading effect. So it was essentially a mismatch between generation and load. The system had been stressed to a certain level, and then a small perturbation led to failure throughout the network. And I thought it was interesting, the comments of a vice president at in the electrical industry. There's conflict between protection of an asset and protection of a system, and you can think of the heart that way. A certain number of cells might decide, well, I want to try to survive this ischemic insult or this metabolic stress, so I'm going to shut myself off. But of course, that's not good for the syncytium and the global aspects of heart function. So we try to understand how failure at the level of even the single organelle, the single mitochondrion, can lead to failure of the energy network, and this is mitochondrial membrane potential in a heart cell, and how that can then scale to affect ion channels and other systems of the cell, affecting the action potential, leading to organ-level dysfunction and fatal arrhythmias. This movie is actually an example of a cell that was stressed to the point of what we call criticality, whereby a small perturbation can cause loss of function throughout the whole network. And in this particular example, which is an extreme example, a single laser flash that depolarized one individual mitochondrion led to blackout of the whole cell. I'm going to talk, and this event is coupled to K peak current activation. What I'm going to talk about today are some of the factors that I think lead up to the oxidatively stressed condition in the context of heart failure, more than this criticality event and the depolarization. But some of the same themes will obtain. So the cascade of failures in heart failure that I'm going to talk about is some of the elements involved in the events leading to sudden cardiac death and congestive heart failure. And the hypothesis is that in pathological states like hypertrophy, chronic heart failure, ischemia, reperfusion, and digoxin toxicity, we have conditions whereby the cytoplasmic sodium levels are elevated, and that, coupled with an impaired cytoplasmic calcium signal, can cause what I call mitochondrial calcium underloading, an impairment of the signaling to the mitochondria that leads to changes in redox potential because there's a mismatch in energy supply and demand, leading to reactive oxygen species, arrhythmias, and cell death, contributing to decompensation. This is kind of the summary slide that you'll see at the end again, and I'll tell you how we got to this, show you the data along the way. But essentially, we're looking at how problem with EC, excitation contraction bioenergetic coupling, can lead to a change in bioenergetic redox coupling and downstream effects on the organ and the animal, whole animal as well. So we were interested in sodium overload of the cytoplasm because we realized in a lot of chronic cardiomyopathy conditions and also diabetes, which isn't shown here, there's an elevation of cytoplasmic sodium. That's very easy to see during ischemia. You have a 4 or 5-fold increase in cytoplasmic sodium that takes a little time to come down when you reperfuse. But also in chronic heart failure models, in a rabbit heart failure model here from Steve Pogwitz, you can see that the resting sodium levels in the cytoplasm are elevated in the heart failure cells from failing hearts. And with stimulation, you get further sodium loading that's exacerbated in heart failure. Also, in humans, this is true as well. Burkitt, Cascade, and Hassenfuss's group published this data in 2002, showing that in this is a lumped together group of failing human heart tissues. They measured sodium, and it's elevated compared to non-failing myocardium. And again, there's a frequency dependence that you get accumulation of sodium at higher pacing frequencies as well. And the idea that I had back in about 2004, 2005, which ended up being a postdoc's project, was that this might impact energetics. And the reason we thought that this might impact energetics is because if we look at the circuit diagram of the ion movements, these are just cation movements across the sarcolemma and across the mitochondrial inner membrane, we can see that the sodium and calcium circuits are linked together across the mitochondrial inner membrane. So there's a sodium-calcium exchange, just like at the sarcolemmal membrane. You have a calcium entry channel, and you have a sodium-calcium exchanger that couples those two circuits together. And in the mitochondria, these circuits are then coupled through a sodium-hydrogen exchanger to the proton circuit and the energy circuit as well. which indirectly then is coupled to the Krebs cycle that's going to control the redox equivalents in the mitochondrial matrix. So you really, although most of you are focused on either the ER or sarcolemmal ion movements, there are also ion movements that are linked to the cations in the cytoplasm in the mitochondria, and these circuits are all tied directly or indirectly to oxidative phosphorylation, because of course all the gradients driving these things are set up by the energy-dependent pumps. So you need energy to set up the gradients and also to couple the flux moving across. And so when I mention mitochondrial calcium signaling, there are good and bad aspects of mitochondrial calcium uptake. One of the bad aspects is that too much mitochondrial calcium uptake can promote the opening of the permeability transition pore, which is the cell death channel. But there are also good aspects of calcium signaling to the mitochondria to maintain energy flow, which I'll get to in a minute. Mitochondria are also a source of ROS, so some of these things are going to affect ROS balance, which is another aspect of what I'm going to tell you today. So mitochondrial calcium efflux depends entirely, almost entirely on sodium. So there's a sodium-dependent efflux pathway that is the predominant calcium efflux pathway in mitochondria. And this shows an example of isolated mitochondria in a cuvette, and it simply shows extra mitochondrial calcium here. So when we add calcium to the cuvette, the signal jumps up, extra mitochondrial calcium is high. Then the mitochondria take up the calcium. And in this particular experiment, we're doing it with 0 sodium outside, so there's minimal efflux. So this is really the unidirectional calcium uptake rate here. It then takes up the calcium, so it's actually lowering the calcium in the cuvette. You can add a blocker of the uptake pathway called Ru360, or ruthenium derivative, and there's virtually no leak out of the mitochondria. There's a very tiny leak, a little bit through PTP, but it's minimal. Then when you add sodium back, you can see that the calcium starts coming out of the mitochondria. So this is the unidirectional uptake, and this is the efflux rate. So it's strictly sodium dependent. This is 5 millimolar sodium. You can inhibit this efflux with CGP 37157, which is a benzothiazepine compound. And it's pretty effective at blocking all of the calcium efflux from the mitochondria. And so 5 millimolar is right around the normal resting level of cytoplasmic sodium in larger animals. It's higher in rat and mouse. But you can see here that the range is really covering the physiological and pathophysiological range that's encountered. So in heart failure, you're up closer to 15 or 20 millimolar. So we hypothesized that changes in cytoplasmic sodium would have a major impact on mitochondrial calcium dynamics, and we wanted to find out what the impact of that was. And this is illustrating that spatial aspects also play a predominant role. If you look at the T-tubule distribution and overlay it on the mitochondrial distribution, this is mitochondrial labeled with TMRM. This is Dyneps labeled hard cell. You can see that the mitochondria are sandwiching the T-tubule on both sides. So we have to be concerned also about the mitochondria. Are they sensing local or global calcium, and how crosstalk occurs between EC coupling and mitochondrial calcium uptake. And if you look at EM, that becomes very evident as well. The mitochondria on both sides of the T-tubule, ranitidine receptors are just a few tens of nanometers away from the release. The mitochondria are very close to the release sites through the ranitidine receptors. The non-junctional SR is wrapped all around the mitochondria. and that makes it difficult actually to get rid of the SR when you're trying to make a pure mitochondrial intermembrane prep. There's always some element that's very difficult to get rid of, and you can see why. As it turns out, there are actually protein tethers between these two organelles. So there's an intimate relationship between the mitochondria and the SR that really can't be ignored.
Brian?
Yeah.
Sorry. Given that, how can you be sure in those mitochondrial preps that it's not SR up there?
Well, we can give fapsigargan or cyclopiezoic acid and make sure that the calcium pumps in the SR are not working, and it actually doesn't affect those fluxes at all. But it is a concern for certain experiments, and we put CPA in there to block any SR calcium uptake. So when I draw my cartoons, I try to keep this relationship between the organelles more realistic and not put the mitochondria over in the corner somewhere where you usually see it on these easy coupling slides. But the idea is that the normal coupling, again, is calcium-induced calcium release, that a fraction of calcium is going to enter the mitochondria through the mitochondrial calcium uniporter. The calcium is going to bind to the myofilaments, and that's going to be the major energy demand in the cell. And just to put it in perspective, you're during when the heart is beating, you're turning over all of the ATP in your heart about every 6 seconds. So you need a continuous flux of ATP production to keep up with demand. So when ADP enters the mitochondrion along with PI and gets phosphorylated, you're actually consuming the proton motive force, which is maintained by NADH, so you're oxidizing some of your NADH. So you need a signal to increase the NADH production to keep up with the demand. So there's a pull on the system, you need some push to go along with it to match the flow. And that signal is thought to be the calcium that's entered during EC coupling. There are calcium-dependent enzymes in the Krebs cycle, or the tricarboxylic acid cycle, that increase the rate of key enzymes in the cycle, which I'll show you a little bit later. But you need that upstream signal to maintain the flow in the system and maintain the balance. As I said and showed you already, the sodium is coupled to calcium efflux. So if you have a pathological situation where cytoplasmic sodium is elevated, this rate is going to increase. You're going to tend to blunt the signal or decrease the calcium signal in the mitochondrial matrix. Of course, sodium has many effects at the sarcolemma as well, and we've studied these in the past as well. When you have elevated sodium, you're going to affect the sodium-calcium exchange, which is going to change the distribution of calcium in the SR that's going to be released, and tends to increase the SR calcium. So you'd be offloading calcium from the mitochondria, distributing more in the SR compartment. The tools that we use are, as I mentioned, this derivative that's part of the conglomerate known as ruthenium red, but this is the more select agent, ruthenium 360, at nanomolar levels that can block the calcium uptake pathway. And CGP 37157, which I'll talk a lot about, which is an inhibitor of the sodium-calcium exchanger. And this project started with a postdoc named Christoph Mack in my lab about 5 years ago. And what we did was develop a way to measure mitochondrial and cytoplasmic calcium in the same cell at the same time. And we used the kind of the classic way. We load the cell with Rho2 AM, which is a dirty signal because you have Rho2 that's in the cytoplasm and then also in the mitochondrial matrix. But then we patch clamp the cells with Rho2 free patch solution, and also our patch pipette contained Indo-1 which dialyzes into the cytoplasm and dialyzes out the Rho2 in the cytoplasm. And we were able to then get 2 different fluorophores with different emission properties and measure calcium in both compartments. And we did all sorts of controls to show that there was no Rho2 left in the cytoplasm and that the signals we were measuring actually were in the right compartments. And this is just illustrating when you increase the cytoplasmic calcium signal with isoproterenol. So this was isoproterenol at 3 different concentrations. The peak cytoplasmic calcium increases, the diastolic doesn't change. And so we get this graded response in cytoplasmic calcium. If we track a mitochondrial signal, it's very small but visible during the very small calcium transients in the cytoplasm. And then there's a transient component that's occurring with each beat. And there's a slow decay. So the uptake is fast and the decay is slow in the mitochondrial signal. And whoops, I hit home by mistake. Sorry about that. This is where I should learn how to jump to a given one. But you'll notice that because the decay of calcium in the mitochondria is slower, you get this accumulation effect of the diastolic signal as well. So there's a transient component and a loading effect during diastole as well.
So are you saying if you stop stimulating, the resting will drop?
It comes back down again. It comes back down a little bit slower than it goes up.
Even in ISO?
Even in ISO, yeah. And I'll show you that in the subsequent slides. And just to show you that these signals are responding in the way we expect them to for the different compartments, when we put 10 nanomolar Ru360 inside our pipette, we blunt the mitochondrial signal. If we put 100 micromolar, it blocks it almost entirely. And yet the cytoplasmic signal is increased. So the mitochondria are a component of calcium buffering that when blocked, you get a potentiation of the cytoplasmic signal. So both signals go in the opposite directions with the uniporter inhibitor Ru360. If we treat the cells with CGP 37157 in the pipette, you can see that the basal amount of calcium is already higher in the mitochondria, and then it goes up much higher in the CGP treated cells than in controls as you grade the cytoplasmic signal with isoproterenol. And yet the cytoplasmic signal is decreased by CGP. And if you look at the time constants of decay of the mitochondrial signal, it's slowed, but there's no change in the cytoplasmic decay kinetics. So we think CGP is relatively selective for the mitochondrial sodium-calcium exchanger, and it's consistent that other people have not seen much effect of CGP on the sarcolemmal sodium-calcium exchanger. And the changes in the 2 compartments are consistent with that. So then we went on to look at high sodium, pathological, quote-unquote, levels of sodium. So we had cells that were either at 5 millimolar internal sodium or 15 millimolar, and we saw that the mitochondrial signal was smaller in 15 millimolar sodium, and the efflux rate, if you normalize these signals, is faster. So we've increased efflux and decreased the mitochondrial signal. And again, the opposite effect happened on the cytoplasmic signal. You increase the cytoplasmic signal. So as I mentioned before, the distribution of calcium is moving not in the mitochondrial direction, but in the SR direction with higher sodium. So we then looked at what happens to energetics, and the way we looked at it was to look at the autofluorescence that's due to the NADH primarily in the mitochondria, and we looked at the NADH reduction state during a work transition. So this protocol is we treat the cells with isoproterenol, but they're not stimulated yet until this point, and then we do 4 Hertz stimulation. So it simulates going from rest to work. And you can see that with low sodium, 5 millimolar sodium, you get loading of the mitochondria with calcium. But at 15 millimolar, you really blunt the rise in mitochondrial calcium. So you're blunting the mitochondrial calcium signal. The calcium comes back down. This is kind of complex kinetics of a decrease. It doesn't jump down suddenly, but it goes down over a slower time scale. But the point of this slide is that in the same cells, we're looking at the NADH response. And you can see at low sodium, you get a transient decrease in NADH at the onset of the increased demand, but very rapidly everything compensates and your NADH levels don't change significantly from the resting state. And when you stop the work, there's actually a bit of an overshoot of NADH because the Krebs cycle is actually geared up to produce more NADH, and now the demand has dropped. So you transiently overshoot on the NADH. However, it's dramatically different with high sodium, and you can see there's oxidation that's not reversed. So you get net oxidation of your NADH pool that corresponds to the less loading of calcium in the mitochondria, so the mitochondrial calcium signal is not as strong. The other thing to note here is that there's no recovery of the NADH, even though the demand has decreased out here. And this was the first indication that we had some irreversible process, which we hypothesized was oxidative damage, and it turns out to be true, which I'll show you. And so there's a deficiency of energy supply and demand balance here, leading to net NADH oxidation in the mitochondria. And this is showing the mitochondrial calcium at 15 versus 5 millimolar and the differences in the NADH response. If we treat the cells with CGP, we potentiate the mitochondrial calcium responses and in fact eliminate the oxidation during the increase in work in this experiment. Conversely, if we use 100 nanomolar Ru360 and totally eliminate the mitochondrial calcium signal, or largely eliminate the mitochondrial calcium signal, you can see the Ru360 treated cells have blocked the mitochondrial calcium uptake regardless of the sodium level in the cytoplasm. And in that case, you get the maximum NADH oxidation effect. So you really need this calcium signal getting to the mitochondria to maintain NADH. Does it have any pertinent information that's relevant to heart failure in vivo? In this study, we applied ascending aortic constriction to create pressure overload induced heart failure in the guinea pig. And then we chronically treated these animals with or without CGP 37157, which was implanted using an Alzette mini pump, osmotic pump inside the animal. And so first off, when we look at 4 weeks after aortic banding, we have significant hypertrophy in the failing group. The heart weight tibial length is elevated. The ejection fraction is diminished. And we measured resting sodium levels in cells sitting in the dish without a patch pipette and using SPFI, the ratiometric sodium indicator. And we showed that in the failing group, resting sodium is as high as 17 millimolar average, whereas in the control group, it's about 5 millimolar. So it's over the same range that we had studied the normal cells. Now, if we just field stimulate those cells in the dish and measure the NADH response, you can see that control cells, their NADH does not oxidize. There's a maintained balance of demand and NADH supply. Failing cells, if you stimulate them, there's this marked depletion of the NADH in the mitochondria. But if you pre-treat those failing cells with CGP 37157, you eliminate the redox changes that happen in the failing cells. So I showed you that the CGP before in normal cells can restore the mitochondrial signaling and eliminate the oxidation of the NADH. And here I'm showing you that in failing cells you get the same effect of CGP. And we wanted to know whether there's an intrinsic problem with failing cell mitochondrial calcium dynamics, or whether it was mainly due to the changes in sodium that we measured. So we took failing cells and we did patch clamp on them and forced them to have lower sodium. So you can see here the comparison in 15 millimolar intracellular sodium. Failing cells have blunted mitochondrial calcium signals that are lower than the normal cells. But if you lower the sodium in the failing cells, you potentiate the mitochondrial calcium signal. There's still kinetic differences because don't forget the cytoplasmic calcium transient kinetics are altered in failure, and we haven't cured that part of the problem with sodium. But what we have done is potentiate the mitochondrial calcium signal here. And you can see when you lower the sodium in the failing cells, you get a better increase in mitochondrial calcium during the increase in work, and you eliminate this NADH oxidation problem as well. So both CGP or lowering sodium manually in your patch pipette are ways of eliminating this problem. Now this, I don't know if the lights could go down a little more. You can probably see it. So this is a summary of all the data I showed you so far, and it's on this axis we see the decrease in NADH, so the bigger oxidation responses are down here, and this is the calcium in the mitochondria at the end of the pacing period. So you can see the worst case scenario, as I already showed you, is when you have Ru360 present, and this is the 15 millimolar sodium with Ru360, 5 millimolar sodium with Ru360. These are normal cells. These are failing cells with 15 millimolar sodium, and these are normal cells with 15 millimolar sodium. So you can see it's starting to fall along this line. If we treat cells, high sodium cells, with CGP, you can see you can go from down here up to here, and you eliminate the NADH drop. So the bottom line here is that there seems to be a threshold of mitochondrial calcium signal that is required to maintain your NADH flow in the system. And if you drop below that, it falls along a line here with a high correlation coefficient. And you have impaired energetics. The other thing that can potentiate your mitochondrial calcium signal is increasing the phosphate. So this is with 0.2 phosphate. This is 15 sodium with 2 phosphate, and 5 sodium with 2 millimolar phosphate inside. You can see, even though we've increased the mitochondrial calcium signal a lot here, it's maintained kind of at the same level. There's no overshoot in this case. So it's really, you just need a certain amount, and that's enough to maintain the system. So I mentioned that we thought there was oxidative damage going on during this high sodium situation and NADH oxidation. So the question arises, how is metabolism coupled to oxidative stress? And so the hypothesis that we came up with is that it's known that the TCA cycle is going to be important for your ROS scavenger pathways. And I point out here that the TCA cycle is stimulated by calcium at alpha-ketoglutarate dehydrogenase, isocitrate dehydrogenase, and pyruvate dehydrogenase. So you need to detoxify ROS, and one of the major sources of ROS in the heart cell is from the electron transport chain. And two of the key sites are complex 1 and complex 3. Complex 3 can leak single electron reduction of oxygen, can cause superoxide to come off at complex 3, either on the outside or the interface of the membrane of the mitochondria. That superoxide has to be detoxified by manganese superoxide dismutase enzyme, which generates hydrogen peroxide. So ultimately, you have to get rid of hydrogen peroxide to avoid ROS overload in the system. And the way the mitochondrial matrix gets rid of hydrogen peroxide is through peroxidases. The peroxidases are all going to depend on NADPH. And there are three main pathways for generating NADPH, and all of them involve either the Krebs cycle or NADH itself. So there's a NADP-dependent isocitrate dehydrogenase that reduces NADP to generate NADPH. There's a malic enzyme that takes malate to pyruvate, generating NADPH. And there's a so-called nicotinamide nucleotide transhydrogenase in the membrane that utilizes the proton motive force to couple NADH to NADPH interconversion. So NADPH has the most negative redox potential of all of the carriers, all of the redox couples involved in the redox pathways that detoxify hydrogen peroxide. So NADPH is used by glutathione reductase to keep the glutathione pool reduced, and that in turn, the glutathione is then used by glutathione peroxidase to turn hydrogen peroxide into water. So you need this master electron donor here to keep this coupled moiety-conserved redox cycle intact. It's also used by thyroidoxin reductase to keep thyroidoxin reduced, and the thyroidoxin is important for maintaining the reaction of the peroxyredoxin 3 in the mitochondria, which is another peroxidase. And there's also a glutathione reductase in there, which I already mentioned, actually. All right. So the question that comes up, if you are at all involved with thinking about ROS production, is why is it that when you do more work, you have more ROS produced by the mitochondria? The dogma is that when electrons back up and you have very low flow and you're not doing any work, that's when you get more ROS leaking from complex 1. Well, I'm not going to get into all the details and the data behind this figure, but in this paper we described how you can actually get ROS at both ends of the redox spectrum. And there's a classical view out there that started with Skulichev and Starkov that showed that ROS production leak from the electron transport rate increases as you get more and more reduced. So it turns out that when NADH levels are very high, you start reducing the site in complex 1 that will transfer electrons directly to oxygen and generate superoxide. So, and that's true in isolated mitochondria. You can get them to generate a lot of ROS when there's a high redox potential. However, we're seeing more ROS on this end of the spectrum when the redox pool is more oxidized. So the explanation for that is there's two components to ROS overflow in the system. So if you're not doing any work and you have a lot of redox equivalents and you're highly reduced, and you can drive isolated mitochondria to this state, so-called state 4, the rate of ROS production can actually ramp up and exceed the level of ROS scavenging in the green trace here. However, when we're in the high work situation, what's happening is we're depleting your NADH and NADPH, and everything's getting more oxidized. So your ROS scavenger pool becomes the limiting factor. So even though you may not have a higher rate of ROS production, your rate of ROS scavenging is dropping. And so you can get elevated ROS overflow at both extremes of the redox pool. And both may be important pathophysiologically. For example, during hypoxia, you reduce your pyridine nucleotide pool almost completely. So you could get ROS by this, in this arm of the pathway. But metabolic demand or exercise or things that cause more oxidative stress, oxidation in the system, can cause ROS to accumulate when you have oxidized redox potential. And that was exactly what we found when we looked at failing heart cells. So if we look, this was published in 2010. This was a collaboration between Christoph's new lab and my lab as he was making the transition. And you see here in our guinea pig heart failure model, the non-failing cells, you can increase their workload in the presence of isoproterenol, just like I showed you before. And if we measure ROS accumulation in the cell, you see that the non-failing cells, the controls, don't have any significant response. But failing cells, which had the elevated sodium, the oxidation of the NADH, have about a 20-fold increase in the rate of ROS accumulation when you stimulate them. But if you treat the failing cells with CGP, you prevent that rise, and they look just like the controls. And just like I showed you the trick with the lowering the sodium in the failing group, that also eliminates the rise in ROS accumulation in the cell at high work. So either lowering sodium or treating with CGP, which both impact the mitochondrial calcium efflux, sodium calcium exchanger, will eliminate the oxidative stress associated with rapid pacing and beta adrenergic stimulation. Just to reinforce that, we also did a study using cardiac glycosides, and I just like to refer back to this painting. Many of you have probably seen it. It's a woman with heart failure and the physician treating her with this extract of some herbal concoction, presumably, and presumably one of the active ingredients of there in that concoction was the foxglove extract. And in 1785, William Withering wrote a paper on the account of the foxglove and some of its medical uses, and he looked at the folk remedies and figured out that the foxglove plant was the active agent in there. And this is the origin of the use of cardiac glycosides like digoxin, digitalis, and wabane. They're all in the same class, which are inhibitors of the sodium pump, the sodium potassium ATPase in the sarcolemma. So wabane should do the same thing that heart failure does, and that is raise cytoplasmic sodium, so we should see some of the same effects on mitochondrial calcium signaling. And this is just showing dose dependence of wabane on the sodium in the cytoplasm. And we used, for most of these studies, 1 micromolar wabane, which elevates the sodium up in the range of 15 to 20 millimolar, like the other experiments. And we could show that, in fact, wabane decreases the mitochondrial calcium signal, and that this signal is restored with CGP treatment. So just like the normal cells in high sodium, the failing cells, wabane-treated cells also have this problem of redox balance, and the wabane-induced NADH oxidation can be reversed by CGP treatment. So this is what I call mitochondrial calcium underloading, as I've already mentioned. This can actually beget cytosolic calcium overload. So this is hard to get across sometimes, is that you have this problem of mitochondrial calcium signaling. It's leading to a lot of oxidative stress in the cell, but that oxidative stress is actually screwing up other subsystems of the cell, like the calcium handling subsystem. And it's known that the ryanodine receptor in particular is very sensitive to oxidation. There's a cluster of reactive files in there, and when they're oxidized, the ryanodine receptor becomes very leaky. there are many targets. Many of the ion channels are also modulated by ROS or sensitive to redox potential, many of the K channels as well. But what we observed in Wabane was that the sodium loading induced by Wabane caused spontaneous calcium releases, for one. These are inhibited by CGP treatment, and accumulation of diastolic calcium when you pace the cells is blunted by CGP as well. So these spontaneous releases were connected to triggered action potentials that were induced by DADs, and those DADs were also suppressed by CGP treatments. This is scaling up now one more level to the intact heart. So these are Langendorf perfused hearts. You treat them with Wabane. There's a little bit of a positive inotropic effect, but there's a lot of arrhythmias that are generated. If you follow the Wabane with isoproterenol, there's often a transition to VF. This is the ECGs on the bottom. This is the volume conducted ECG in the bath around the heart. If you treat with CGP, you prevent the arrhythmias induced by Wabane and by isoproterenol, and you also potentiate, you can probably see the forces is improved as well, in part because you've reduced the synchronous electrical activity. This is just the arrhythmia scores from that study, so you're significantly suppressing those arrhythmias with CGP. There's a positive inotropic effect of CGP, a small one at baseline. It really becomes more prominent after Wabane treatment or Wabane plus isoproterenol. You can see the positive DPDT in the hearts is dramatically potentiated by CGP, and the incidence of VF was significantly decreased. So we think that part of digoxin toxicity might be due to this bioenergetic problem, and we would expect that it could be maybe prevented by CGP treatment and with improvement of the positive inotropic effect. This is just showing that if we measure ROS in the same system, the Wabane-treated cells behave like the failing cells, that there's a very high rate of ROS production, ROS accumulation in the cell with pacing, and CGP prevents that increase in ROS that's happening at pacing. So now, does it work in intact animals? That's the big question. So we looked again at aortic-constricted guinea pigs. This time, though, we changed the model a little, and we lessened the constriction slightly, but we added on top of that chronic catecholamine stimulation. So these animals, after they were banded, they received 1 milligram per kilogram dose of isoproterenol injected once a day into their peritoneum. And they were either treated with or without CGP simultaneously with the osmotic pump. So the CGP is there all the time. The isoproterenol is 1 shot a day. So the first week was 1 milligram per kilogram. The subsequent weeks after banding were 2 milligrams per kilogram, which is not an extremely high isoproterenol dose. It's modest. So in this model, which we're calling the ACI model, we see that there's dilation of the heart at 4 weeks, that there's a decrease in ejection fraction, or fractional shortening here from the echoes, and that with chronic treatment with CGP, we have improved fractional shortening. This model is a hypertrophic model. The banding plus ISO, there was an increase in heart weight to tibia length. This, actually, I forgot to put the star on here, but there was a slight a decrease, but it was significant with CGP treatment in the heart weight increase, so it's inhibiting hypertrophy somewhat. But it's not completely eliminating the hypertrophy. I should mention that we also did the control, the sham plus the isoproterenol treatment without the pressure overload, and they didn't have the impaired contractile function. but they had the catecholamine stimulation. This is PV loop analysis showing the sham and the sham plus daily isoproterenol. You can see the effect of the isoproterenol was to steepen the end-systolic pressure volume relationship. The banded plus iso had impaired systolic and diastolic function, but CGP treatment prevented the heart failure associated changes. So improved contractility. Now we're going to look a little bit at the electrophysiology in these animals. So this is the effect of isoproterenol. This is the daily one-shot injection into the peritoneum. And this is the first week after banding, and this is the fourth week after banding. So this is sort of the compensatory hypertrophy phase and the decompensated phase. And this is just showing the heart rate response. So we see that there's an increase in the heart rate after the isoproterenol injection, no surprise. It decreases over 2 to 3 hours. There was no real difference in the different treatment groups in this heart rate response. They all had the same maximal response. The kinetics look the same for the recovery as well. At 4 weeks, the same story. The 2 milligram per kilogram dose, you had heart rate increasing and then decreasing again. The only differences really were that in the banded plus iso group, there's a bit of a elevation of the baseline heart rate that was significantly, but it's not a huge effect, but there's a decrease here with the CGP treatment in the basal, but the maximal isoproterenol effect was the same. And again, over here at 4 weeks when the hearts are more failing, you see the basal heart rate was decreased by the concomitant CGP treatment. What we did observe, though, which was striking, was that by 4 weeks, we had almost 60% of the animals had died. So we hypothesized that this was sudden cardiac death, but we had to prove it. So in subgroup of the animals, we implanted telemetry so that we could monitor the electrical events. And so at the top here, you see the banded plus iso. This is animal 4 weeks after banding. This is the baseline. This is 10 minutes after the isoproterenol injection, so you see the heart rate increased. This is approximately 3 hours after the isoproterenol injection, and you can see the heart rate is slowed down, but you start to see some PVCs here leading to VT, and then this animal died at that point. So we think that they're dying of sudden cardiac death during this period and not pump failure. What you can see here is that if we treat with CGP, we don't see these electrical abnormalities. You see the increase in heart rate and then the recovery. And basically none of the animals died after the first week after surgery, which was similar to the sham operated plus isoproterenol. So I think there's a post-surgical effect here where isoproterenol is probably, they're not tolerating that very well. But you can clearly see that the failing hearts are not doing well and the CGP animals are surviving. This is just a more detailed analysis, and I'll wrap it up, so I know it's getting late. This is showing the ECGs. This is the first week after the aortic constriction, and this is the baseline ECG in the sham. This is the peak of the heart rate response and then the recovery. And you can see these premature ventricular beats. This is the banded animals, and you could see this one, we captured the transition to VF, or this looks like VT, maybe not VF yet, but this animal died as well. And then the CGP treatment, we have some PVBs, but not a whole lot going on here. This is the quantification at baseline before the isoproterenol injection. The banded animals that didn't receive CGP had a bit higher premature beat index, and then after the isoproterenol, the the PVBs were very high in the heart failure group, and that was blunted by the concomitant CGP treatment. Then this is 4 weeks after banning, the same sort of pattern happening, and you could see the CGP has really prevented the increase in PVBs, and obviously the animals survived, so we've also prevented the VF in this model. This is the QRS duration and the QT interval. There's a bit of a lengthening of the QRS complex that occurs also in the CGP treatment group, but not to the same extent. The corrected QT interval here is also elevated in both of the banded groups. No significant suppression by CGP. That's week 1, and this is week 4, where everything sort of looks similar here. And this is just showing fibrosis is also blunted in the CGP treated group. So the bottom line is, as I started out, high sodium, probably combining with the impaired calcium signaling in the cytoplasm in heart failure, is going to lead to an impairment of the mitochondrial and bioenergetic signals that lead to a change in redox potential that compromise ROS scavenging and contribute to triggered arrhythmias, DADs, and cytoplasmic calcium overload. Ultimately, that can also contribute to sudden cardiac death. And we also believe that more cells are going to be dying by necrotic and apoptotic death due to this chronic adaptation or maladaptation. I throw CAM kinase in here because I know Urim is part of the Leduc to study CAM kinase and has modeled CAM kinase, but there are other elements of this vicious cycle here that can exacerbate this problem, one of which could be the autooxidation of the calmodulin kinase 2, which leads to persistent activation of this kinase, which can contribute to more late sodium current, so-called late sodium current, which is thought to contribute to the leak of sodium across the sarcolemma, which would feed back, obviously, on this loop. Also, CAM kinase may be affecting other ion channels and calcium handling that might exacerbate the arrhythmia part of this as well. So finally, I'll just show you the picture of the group. Christoph Mack is not on here, but Ting Liu did almost all of these studies, Wabane, the heart failure model, and the rest of the crew is working on various other projects in here. So, them by name. So, and I thank the NIH for their support as well. So thank you very much.
Okay, we know everything about the mitochondria now, so this is open for discussion and questions.
So superoxide, if it's not disproportionated into hydrogen peroxide rapidly enough, will turn into singlet oxygen. Have you ever looked for singlet oxygen mediated damage in the mitochondria or during different ROS promoting circumstances?
No, we haven't, but we think the superoxide is probably the mediator of mitochondrial depolarization, which I really didn't talk about. Like,
that was going to be my next question.
These acute changes in high ROS, they're not immediately leading to mitochondrial depolarization. And we haven't really connected this story with our other story, which is what happens when you get chronic oxidative stress and they become susceptible to depolarization and you get KHP current activation. So there's some pieces missing between those two stories, but and the superoxide, I think, is the signaling agent that's talking to the neighboring mitochondria and causing the spread of the depolarization when you do reach this threshold for ROS-induced ROS release in the mitochondria. And I didn't talk much about ROS-induced ROS release. I think what we're studying here is kind of the conditions that are leading up to the oxidative stress, which is probably exacerbated by the catecholamines as well. So it's interesting to me that the animals are not dying at the peak of the heart rate response, the catecholamine response. It's some hours after the isoproterenol. So I think that might be a sign that you've induced this chronic stress, this oxidative stress, that then triggered a change in the substrate later on that caused the fatal event. But again, I didn't connect the dots between the two different projects in my lab at this point.
So do you think that the superoxide is then oxidizing sulfhydryls and ion channels or some accessory?
I think it's more the hydrogen peroxide that's maybe, and the hydroxyl radical are the more damaging and longer lasting. But I think the superoxide is doing local signaling between mitochondria and spreading the depolarization signal. Because as you know, but probably no one else, we think that there's a ROS-activated channel that's opening in the mitochondrial inner membrane that dissipates energy, dissipates the membrane potential. We call it the intermembrane anion channel because it's sensitive to benzodiazepines. And that's, we think it's superoxide that's the messenger in triggering that, because we can put SOD mimetics in there and they will block that problem of ROS-induced ROS release. And you would think that would generate more hydrogen peroxide. But I wouldn't bet my life that it's superoxide as the messenger, because when you have a positive feedback loop, ROS producing more ROS, if superoxide is the trigger and you're lowering that, you're also not producing as much of the downstream ROS as well. So this is going a little bit away from that story, but it's relevant, I think. So we think you could see more triggered activity in DADs early with this type of Wabane treatment or whatever, but we think probably the substrate for reentry might be this other mechanism where you have mitochondrial depolarization and gradients of repolarization due to K to P currents, but we haven't proven that yet in the heart failure model. We know that happens during reperfusion, but this is not ischemia reperfusion. Yes?
So obviously the mitochondria is related to the energy consumption. And like you said, very nice data shows once we do the exercise, the ROS will be accumulated. So, interesting to me is I wonder if we do the exercise or emotional condition, like we need more energy or ATP, does the mitochondria, like the normal or just like you said, heart failure, the mitochondrial activities will be changed, or and also the ATP level at normal, or like the heart failure, once we do the exercise or more demand more ATP.
Usually people that have heart failure don't do exercise very well
right.
either,
Uh-huh.
and it can precipitate arrhythmias. So I think maybe the same type of mechanism might be occurring, and they do have exacerbated markers of oxidative stress. But this is the kicker is I don't think necessarily that if you're compensated and you're doing well, and even though you have heart failure, you may not see any intrinsic difference in ATP levels or things like that. It's only when you have this extra stress and you drop that threshold, you drop below that threshold that I showed you, that you might then get all this ROS accumulation. And it could be exercise that induces it. We didn't try to swim the guinea pigs or anything like that. But we just did the isoproterenol injection as kind of like a sympathetic drive, acute sympathetic drive. But I would maybe anticipate that if we did swim the animals, they might drop off as well. It's a good thing to try, maybe.
So I think you've shown very nicely that this compound kind of prevents decompensation and some of the sequelae with heart failure. What about if you already have heart failure?
Yeah, I asked Ting to try that, but we haven't done it yet. If we can reverse the heart failure state with the CGP. Yeah, I don't know. It's a great question.
Is CGP a potential clinical agent here, or is it have some other effects?
Well, the main thing is getting a big drug company to develop this compound that's been around for a long time, so there's no patent protection on it anymore. But we're still trying to look at avenues for developing it clinically. There has never been a full toxicology study on it, for example. It was developed, I forget what year it was developed, but it was developed among a lot of other compounds that blocked. They were looking for calcium channel blockers at the time, benzodiazepines and things like that. This is a benzodiazepine. It didn't have an effect on the ion channels they were looking at, but they did subsequently find that it affected mitochondrial sodium calcium exchanger. But in the meantime, it wasn't developed clinically at all, as far as I could tell from the people I talked to at Novartis, who now owns CBA, but it's hard to track down originally who was working on that compound. But we are trying to figure out ways of maybe doing toxicology and getting it to the next level. Because it seems to work fairly well in guinea pigs. What does that mean, though? At least guinea pigs are closer than rats and mice. That's my little shot at the...
Very nice presentation. Your increase in potassium concentration, quite remarkable. Is it was also a change in concentration of other ions, like chloride, potassium,
We didn't look at other ions. Usually those other things are more tightly regulated, but I wouldn't anticipate that there'd be a huge change in chloride or potassium. When you inhibit the potassium ATPase, maybe. Colin, do you have any comment on that?
intracellular, extracellular.
We didn't look, so I don't know. I'll write that down. Changes in other ions. Yeah.
What are the mechanisms involved in the fibrosis of fat? And
Oh,
does the
someone
redox,
asked me that the other day. And
does the redox thing happen in myofibrosis?
I'm not sure. I mean, it's theoretically possible, but the fibrosis is just used usually as a marker of, hypertrophy, and it could be the fact that we're improving the load situation overall, so that there's less stretch and things like that. So, because the contractility is a lot better, so maybe there's less fibrosis because you're not getting as much stretching and signals going to the.
But then that implies there's kind of actually reversal of that, related to Mark's question where, then once you build up this.
Oh, can you reverse it? Yeah.
Fibrosis that.
Yeah, but since you're the second person that asked me in 3 days about whether fibrosis is, there's some direct effect, that it sounds like something we should follow up and look at. But the reversal question would maybe address that issue too. Yes, Nadh.
Some calcium question. Your heart failure model, did you see any structural changes in myocyte, like T-tubulus modifications?
I should probably not comment on that. I have my opinion of the whole T-tubule story, and I've been looking at them for a long time, is that there's a lot of variability induced by cell isolation. And you can have bad cell preps in a normal or bad cell prep in a heart failure, and you'll get all sorts of derangement of the T-tubules. So back in the early 2000s, I was imaging a lot of cells from the heart failure dogs that we have, tachypacing-induced dog heart failure model. And I came to the conclusion that there's too much variability based on how good the prep was or not, and it was about equally likely that you'd have a bad normal prep or a bad heart failure prep. So, and so I attempted to image them in a perfused wedge in the wall directly, but the dog is such a large animal; it has a lot of connective tissue, and the visceral pericardial cells and everything prevented me from getting good quality images. I could see some T-tubules, but I couldn't quantify it. So my conclusion was that I couldn't prove to myself that the T-tubules were altered in the failing compared to the normal based on the isolated cells, and that one had to look in the intact heart. Now, some people are looking at intact heart models now and see some derangement, so I may revise my view. We also got a bunch of heart failure cells from human hearts from Steve Hauser's group, and we couldn't pick up any statistically significant difference using a 3D analysis, a 2-photon microscopy, and doing Euclidean distance mapping of the T-tubules. There was no significant difference in failing versus normal human heart cells, which it took us like 10 years to get that published in a crummy journal because nobody wanted to hear it. But anyway, so my conclusion is maybe there's structural remodeling of the T-tubules. I couldn't prove it 10 years ago. It may be getting proved now by in vivo imaging more so. So I can't comment on whether I think it is or not. If there is, it will affect DC coupling. No, well, I should go back 2002, I guess. We published a couple of papers, one where we looked at the intrinsic coupling gain, the EC coupling gain. And in the heart failure model, which has really deranged calcium handling, slow transient, small amplitude, we measured SR calcium content. It's like 30% of normal in those cells. If we fix the SR calcium load, what we did was an experiment where we paced the cell in 5 millimolar external calcium, then jumped back to normal calcium, and then looked at the EC coupling gain when we've restored the SR calcium load. No difference in the intrinsic EC coupling gain. So my take-home message from that is if there was structural remodeling, you should have seen it in that experiment. And we've also restored SR calcium load by inhibiting the sodium-calcium exchanger in the sarcolemma. Again, it restored function, and there was no intrinsic structural difference. Now, that doesn't totally rule it out that we killed off all our cells that had bad T-tubules, and we only studied the ones, but at least the ones we were studying had impaired calcium handling, but apparently, in those experiments, there was no evidence for structural remodeling.
Well, there was no evidence that if there was structural remodeling, it had any effect on EC coupling. Is that what you're saying?
Right. Well, the prediction would be if you have a large derangement of the dyad or T-tubules, you would not get the same coupling gain if you fix the SR load. It would still be impaired. And we didn't. But, when you isolate cells, it could be that all the ones that had deranged tubules died off for whatever reason, because I think that's also an oxidative stress.
It's also probably depending
issue.
on your model of heart failure, not all the
Yeah,
same.
yeah, except other people in the same model were reporting all these derangements of T-tubules that I wasn't seeing, and so I don't, the jury's still out, I think, but I think it's possible, especially...
It's also possible that there are smaller changes than this, ultimate derangement of T-tubules, and
Right.
small changes could really be amplified and cause major changes
That's
in calcium
true.
cycling. You don't have to have...
That's
you
true.
reduce the synchrony of calcium cycling by reducing,
Yeah.
for example, or increasing the distances between that calcium channels and random receptors.
Yeah.
That by itself could cause... So
Yeah.
you might not see it
Right.
in your imaging technique, but it could be there
Sure,
functional.
sure. And the other thing I should point out is all the studies we did in the 90, 98, 99, 2000, whatever, we only used the mid-myocardial cells. And so basically, we're still studying the same model. I'm on the CAS-PPG on resynchronization as well, looking at mitochondrial function. But I got out of the T-tubule business because it was frustrating. But Gordon has been sending cells from that model to John Bridge, who's analyzed it, and he sees all sorts of derangements. I'm still skeptical because I studied the same model and didn't see them.
Well, the way
But
to
he's
solve
also
that-
looking more carefully at different regions like anterior wall, septum, and we do see really big differences in gene expression and mitochondrial function in different parts of the
It's very
wall.
it's very
So
regional.
it's very regional, and even those T-tubule structural changes that he's showing in these dogs. It's only in one part. I think the anterior, might have been the anterior wall, but and the anterior wall is not where the maximum strain is. But I don't know.
But I hear more often now that people find more effects on the anterior wall than other parts of the heart. It's interesting.
Yeah, it is, and it's a little strange because it's
It
not
is.
where you would think they're being stretched the most. But you don't know about the forces going on there. Depends on the fiber orientation and everything too. Yeah.
You mentioned there's like a 30% of loading of SR in failing cell compared to normal.
Yeah.
But there, you also seems to, in one of your slides you're saying there's a sodium accumulation is like a lot higher in the failing monocytes. So why is, because I assume if a higher sodium is gonna,
Right,
have
you would think. Yeah,
I
you
would
would
think, yeah,
think.
it's
Yeah,
very-
yeah, that's the opposite effect. It should have the opposite effect.
Right, I just, what's the reason for that?
Right. What happens is during the action potential, you're actually getting more outward current through the sodium-calcium exchanger in the failing heart cells because the action potential plateau is higher, the calcium release is smaller. So actually, there's more reverse sodium-calcium exchange during the active part of the cycle, during the action potential, and that's why you get these small amplitude calcium transients that have like this delayed rise very often. And so you shift that reversal point of the sodium-calcium exchanger. So it's actually bringing in calcium late, but the circa pump is so impaired that it's not able to pump it up into the SR very well. It's really the circa impairment that's more important.
That's the
Yeah.
Yeah, that makes more
So
sense.
there is
You
maybe
think
more
uptake into
reverse
this,
sodium-calcium exchange, but the circa problem is the bigger problem.
Right. Yeah, that makes sense.
Thank
All right,
you.
well, thank you so much.
I'll see you in a little bit.
Yeah.